Memming, you're handling the introduction, is that correct?
I shall try.
Excellent, good. Okay, good.
All right, so welcome everybody. It's my pleasure to introduce my colleague, Dr. Braden Brinkman. He's an affiliate member of IACS and assistant professor in the Department of Neurobiology and Behavior. He got his PhD in physics from UIUC. He's a statistical physicist, as far as I recall, and he has done his postdoc at University of Washington. And he studied many different things in terms of neural circuits and optimality conditions. He's done a lot of impressive work in terms of studying large-scale neural networks and their analytical properties. And today he's going to talk about evolution of innate behavioral strategies through competitive population dynamics. Please virtually welcome Dr. Braden Brinkman.
All right, thank you for the introduction, Memming. So today, what I'm going to be talking about is not quite down at the circuit level in brains yet, but is a line of work that I'm hoping will eventually start incorporating some aspects of neural circuitry and how we process sensory information. But we're going to start at a little bit higher level than that, which is looking at the dynamics of populations and how evolutionary selective pressures might pick out particular types of patterns of behavior in these populations. And so today I'm going to be talking about work that I've been doing with my graduate student, Tong Leong, from the physics department. And so let me jump into it to sort of set the scene. So foraging is a fundamental problem that organisms have to deal with out in the wild. So imagine, for example, that you're this schematic cartoon snail in some environment with mixed distributions of patches of grass. So currently it's in a position where, the grass is fairly lush, pretty green, somewhat nutritious, perhaps is a decent match to the color of the organism, providing possible protection or camouflage from predators. And this organism has a fundamental problem that it needs to solve, which is, should it stay in its current location and just keep exploiting the resources that it has where it is, or should it take the risk of exploring its environment in hopes of finding some places where the grass is greener, so to speak, at the risk of encountering places where perhaps the grass is not so green. And it has to do this with a sort of limited view of its environment, represented schematically here by the highlighted portion of this circle. So the snail can only sense within some vicinity of its body, and so it has to make an instinctual decision in order to venture off into the unknown or stay where it is. And so if it decides to move, perhaps it will get lucky and find a patch of grass that is indeed lusher, more nutritious, perhaps a better match to its color, providing better camouflage. And if that happens, that's going to be a good thing for the snail. It's sort of improved its station in life, potentially giving it more ability to go on and have many snail babies and propagate its genes. However, if the snail happens to get unlucky and it moves into a location where the grass is now dry, not nutritious, perhaps it stands out more against this grass, then it's at risk of, not getting enough food to reproduce, or maybe even being eaten by a predator. And so this would be a sort of bad outcome for this organism. And, so the organism basically has this problem that it has to figure out, should I stay where I am, or should I take the risk of venturing off in hopes of finding more resources, more opportunities to proliferate? And these organisms don't get taught what the best sort of foraging strategies are by their parents. The snail parents are not teaching snail babies how to navigate an environment. This is all something that, or for the most part, has been sort of hard coded through millions of years of evolution. And so the the view that we want to take in this modeling problem is trying to understand what the selective pressures are on different kinds of foraging strategies for organisms in uncertain environments. So the sort of primary question we're going to be tackling today is, which of these behavioral foraging strategies does evolution select for, and how do those strategies depend on the statistics of resources within the environment? And so again, this is the sort of primary question that we're going to focus on today. But we do have a deeper question in mind with this line of work that we hope to start tackling, which relates to some of these questions about optimality that Memming mentioned briefly in his introduction. And so one of the higher level questions that we're keeping in mind here is, can we derive what's called an evolutionary fitness function that will predict what these most successful strategies are? And to just unpack this second question a little bit, it relates to this idea that's prominent throughout mathematical biology of this notion that biological creatures may be in some sense optimized for their environments. And many of you are probably familiar with this notion of survival of the fittest, that this idea that was put forward that the organisms that are somehow most fit for their environments are the ones that are going to go on and to survive and proliferate. And what this has led to in terms of mathematical modeling of biological systems, at least a popular approach, is to view evolution as a kind of optimization process. And so typically what you see in mathematical modeling is one defines some kind of fitness function or fitness landscape, depicted here schematically, where the organism's fitness, so its fitness for surviving and proliferating in its environment, is some function of the organism's traits, which here we're going to take to be parameters that affect the organism's behavior or its phenotype. So today I'm not going to be talking about evolution at the level of genetics, where I'm going to be focusing on the selective pressures on the behaviors of the organism. So those set of behaviors I'm going to refer to as the phenotype. And in the modeling I'm going to talk about today, the phenotype is going to be dictated by a set of parameters that I'll explain for our specific model in a few slides. But the idea is that the fitness of an organism, or really a population of organisms, will vary with these phenotype parameters. And it could be something bumpy looking like this, where there are certain combinations of parameters for which the fitness is sort of locally at a peak. And this idea of evolution as an optimization process says that evolutionary selective pressures are going to basically select for organisms that tend to maximize their fitness. So if we imagine starting with a population of organisms that have a spread of these traits in the environment, what these evolutionary selective pressures are going to do is sort of select for those traits which are located near these local maxima of this so-called fitness landscape. And some of the surviving organisms may only converge to local maxima, while others might converge to this global maximum depicted here in the center. And so this idea has given rise to a few different types of approaches in mathematical modeling. So a very common and pragmatic one is to define some kind of cost function that we view as a proxy for an organism's fitness. So for example, we might assume that organisms ought to try to maximize the information they have about certain features of the environment, or we might assume that organisms should be able to perform a particular task, and this cost function basically imposes a penalty for deviating from good performance on the task, and then we would select parameters that optimize that cost function. So that's a very common approach that we see in mathematical biology and computational neuroscience. And for the most part, it's a very pragmatic approach and has been successful because we don't want to always be, running simulations that are actually trying to evolve these things. But this is a more sort of abstract level of approach to trying to understand the features that biological organisms tend to have. A slightly an approach that sort of moves more in the direction of dealing with the evolutionary dynamics might be to define a model with a single representative agent, or perhaps a population of agents being the organisms, that share a common set of phenotype parameters. And then what you would derive in such a model is the growth rate of the population, or perhaps the expected survival time, which will be a function of the parameters of the model. And then you find the parameters that optimize or maximize the growth rate or the survival time. The assumption being that evolution would again select for those parameters. However, what this approach leaves out is a possible interaction between organisms with very different phenotypes. And that's the approach that we wanted to focus on with this work is, what if we have a population that has a distribution of different phenotypes where they are competing for resources? And in this case, it's not obvious that that there is a fitness function that's just a function of one arbitrary set of phenotype parameters. In general, it could be, all the phenotypes are there to begin with. And so in some sense, what happens in such a model down at the bottom here is that you have an initial distribution of phenotypes, and then after some length of evolutionary dynamics, you end up with a different distribution of phenotypes. And so there's a sense in which the fitness function isn't something that you define a priori and maximize, but it's just something that emerges from these evolutionary dynamics. And that's the approach we wanted to take to investigating this foraging task, with this longer term view of extending it to other kinds of models where we can perhaps start to try to understand how that kind of approach relates to these more abstract methods where we define a cost function that's a proxy for the evolutionary fitness. So this is sort of the higher order objective that we have in mind and are sort of, aiming to try to start studying with this more simplified setting, where we're going to start with looking at these problems in foraging in random environments. And we're going to take a few different approaches to trying to understand that. The first is going to be a set of agent-based simulations, which I'll describe shortly. And then we're going to try to understand the outcomes of those agent-based simulations using some deterministic mean field modeling. And then the last thing that I'm going to talk about today is pushing these simulations and these models into a high density regime. So where the environment is very densely packed with organisms, to investigate how that might change these most competitive foraging strategies. So I'll move on to the agent-based population dynamics, and that will make some of the setting more precise and hopefully clear. Excuse me. All right, so the setting that we're going to work with is we're going to be looking at the dynamics of agents in a 2D random environment. So we're going to model this as a 2D grid, as shown here, where each location in the grid has a different resource level depicted here by the colors. And those resources are randomly distributed throughout this environment according to a certain frequency. So in the example shown here, we have 5 resource levels, and they are all equally distributed throughout this environment. We're going to assume no spatial correlations between these resource levels. So the resource level at one site is uncorrelated with the resource levels at any subsequent sites. And you can think of this as sort of an extreme hard limit of this type of environment. So an organism in this environment isn't going to be able to learn information about spatial correlations in the environment that will help it navigate it. And I should stress that I'm going to call these resource levels, but these resources are not consumable. So you shouldn't think of them as food, but perhaps more so as something like temperature or shelter, where the different levels are essentially indicating different birth rates that occur at these sites. So each of these resource levels is going to be assigned a particular birth rate in this environment. So resource level 5 will have the highest birth rate, equal to 4 in some units in this example. And then the lower resource levels will have lower birth rates, such that resource level 1 sites in yellow here have little to no birth rate there. So an organism at a dark green site in this environment would have a high birth rate, whereas an organism at a yellow site would essentially have no birth rate. And so what we're going to be investigating is the dynamics of these organisms on such an environment. Excuse me. All right, so let me explain what those dynamics are. So each organism in this environment is only going to be able to detect the resource level at its current position. So for example, one of these snails in the lower left-hand corner here, they can tell they're at a site of resource level 1, but this particular snail cannot tell that there's a site of resource level 5 right in front of it. So it has to move basically just based on the information of its current location. And the way that it can move is that it's going to be able to randomly hop in one of the cardinal directions here, so to a nearest neighbor site, at a rate that is a function of the current resource level. So there are 5 resource levels in the simulations I'm going to be showing you today. So each organism has a set of 5 different hopping rates, 1 for each of these resource levels. And so that's going to comprise the phenotype of the organisms in this environment. So it's going to be a 5-dimensional phenotype that is just the set of their hopping rate out of sites of these different resource levels. And we're going to constrain the values of those hopping rates to be between 0 and 10 in some arbitrary units, where 0 represents that the agent is never going to move out of a site of that particular resource level, and a hopping rate value of 10 would indicate that the organism is going to try to leave that site at its maximum possible rate. So there's a maximum rate at which an organism can leave a site, and a minimum rate which represents it just staying where it is indefinitely. And so, for example, if we pick one of these snails and we randomly assign it a phenotype, that might look something like this. So the phenotype as a function of resource level, and you see that this particular snail will leave sites of resource level 1 and 2 at a middle rate of about 6. It's going to leave sites of resource level 3 pretty slowly at a hopping rate of 0.5 or 1 or so. And then it will leave sites of resource level 4 and 5, the darker green sites, at a pretty high rate. And so just to animate schematically what that might look like, so the snail will make random hops through its environment, and you see it moves through the darker green squares pretty quickly, yellow and light green at a middle rate, and then it's going to leave this middle square at a very slow rate till it gets to a dark green square when it moves quickly again. Excuse me. And again, in the real simulation, all of those hops will be random in continuous time. So they're not going to be quite as smooth like that, but this was just to represent the speed with which it might move through the environment. And of course, all of the snails depicted here are going to have their own phenotype, their own random phenotype, displayed schematically in this plot here. So each one will move with its own different set of rates through the environment. Excuse me. Okay, so that's how the organisms will move through the environment. But they will also be able to give birth, because as I mentioned, each of these sites has an associated birth rate. And we're going to assume that for simplicity, birth in this model is asexual. So a single parent can give birth to a single offspring, and those births are going to be given into adjacent sites. So if this snail here that we just saw move through the environment gives birth, it might give birth into this site here, where now this circled snail is the offspring. And the offspring in this model are going to inherit a noisy copy of the parent's phenotype. So the parent here was the snail with the bolded hopping rate curve in this plot. And so its offspring's phenotype might look something like this. So a slight perturbation of the parent's phenotype. And this is going to be the extent to which there's any kind of mutation in this model. So the children receiving a noisy copy of the parent's phenotype. Excuse me. All right. So this is how the snails can move and reproduce. But the last ingredient we need is some kind of selection event, some form of death in this population that's going to be able to essentially separate the more successful organisms from the less successful organisms. And we're going to implement that by a set of periodically recurring catastrophes that will reduce the population randomly back to its original size. So in this cartoon, we originally had 7 snails. There's been 1 birth. So this catastrophe would randomly select one of the snails in the environment and kill it off, removing its phenotype from the plot here. So one of the phenotypes disappeared because we lost the snail with that phenotype. Now, of course, this sort of catastrophic event isn't the most naturalistic way that organisms die. So you can imagine this as in terms of an experiment where we're growing a population of organisms in a lab over some set time period, maybe a week or so, and at the end of the week, we randomly cull that population back down to its original size. And this catastrophe is going to result in selection because the organisms that are able to reproduce more between these catastrophes are going to be more likely to survive because they'll just have more individuals with similar phenotypes, so they have a better chance of surviving the catastrophe. So that's how selection of the phenotypes is going to occur. And what we want to investigate is which phenotype is actually going to confer the greatest reproductive success. You might anticipate that this example phenotype that I've shown here is probably not the best that this population can do. And if you think about it, you can probably come up with something that is intuitively better. So as I mentioned, these dark green sites are the ones with the highest birth rate. So it seems like what might be a better strategy is for these organisms to just leave any site that is not the highest resource level as quickly as possible. And then when it finds a high resource level site, it should just stay there. So we might intuit that the optimal hopping rate function would look something like this. So maximum rate at sites 1 to 4, and then minimum rate when it finally gets to site 5. Excuse me. And now we're actually going to go from the cartoon to the simulations to see the extent to which this intuition actually turns out to be correct. So is everyone with me so far? All right. So
Yep.
So now let's go take a look at the actual simulation results. So now on the left here, what you're seeing is a 128 by 128 environment where the shades of yellow to green, again, represent the random resource levels. And you can hopefully see scattered throughout the environment some colored dots ranging from cyan to magenta, which represent the organisms distributed throughout this environment. And this color scale corresponds to the hopping rate that the organisms have out of resource level 4 sites. And we'll see why we've colored them with that color momentarily. And then on the right-hand side here, what you're seeing is the distribution of the different phenotype components in this population. And so this is a violin plot. And so what each of these violins represents is sort of a histogram smoothed out on its side. So for example, this very first column is just a histogram of the values of hopping rate 1 that occur throughout this population that's been smoothed. And so we're starting with basically a uniform distribution of hopping rate values in this population. And then this distribution is going to change as we run these evolutionary dynamics. And so the first thing that I'm going to show you is the dynamics from the start up to the first catastrophe so that you can see some of the individual hopping and birth events. So I've zoomed in here so you can see it a little bit more easily. And then when I play this, you'll see some of these dots move and some dots appear, which represents birth events. So if we take a look at that, you can see that some of these dots are moving. Sometimes you have a dot that appears representing a birth, and they have about 0.3 seconds in these or not 0.3 seconds, but 0.3 time units with which to move around and give birth before this catastrophe occurs and culls the population back down to the original 500 agents. And this distortion here is not a representation of the catastrophe. It's just something funny that's happening during the screen share, but you can maybe think of it as the random catastrophe that's culling these agents back down to the initial size of 500. And now what I'm going to do is I'm going to show you snapshots of the population after each catastrophe. So after it's been culled back down to 500 agents, and you'll see how the distribution of the population changes with each of these catastrophes. And so, I recommend focusing on the left-hand plot so you can see the shift in color of dots on the plot, and we'll focus more on the distributions on the next slide. So I'm going to play this now, and you see that the dots started off with a distribution of colors, but then they appear to get pretty uniform. But then the shade of the color of the agents on the left-hand side seems to be slowly shifting in time. And at the end of the simulation, most of the colored dots here are between purple and magenta, indicating that most of these organisms have hopping rates gamma 4 between 8 and 10. And I should mention that this simulation is one trial, whereas these distributions on the right-hand side have been averaged over 20 trials of these simulations. All right, so if we now go and look more closely at how the phenotype distribution evolved over 50, 500, and 20,000 catastrophes, we see that the evolutionary dynamics do seem to be selecting for organisms that have high hopping rates in sites 1 to 3, a very low hopping rate in site 5, and then reasonably high hopping rates in resource level 4, but with perhaps a longer tail of the distribution. And we can also look at how the averages converge in time across these 20 trials. And we see that hopping rate 5 converges to 0 pretty quickly. Hopping rates 1 to 3 also converge, saturate at a high value pretty quickly. Whereas hopping rate 4 is maybe saturating, but there's a lot of variability there. And so this mostly agrees with the intuition that I mentioned a few slides ago, that we think that the best thing these organisms could do is hop at high rates out of any site that's not the highest resource level, and a low rate out of sites that is the lowest resource level. But it turns out that there seems to be a much weaker selection pressure on the hopping rate out of sites of resource level 4, the second highest hopping rate. So we end up with a wider distribution. So it's not as strongly selected for. And so this naturally leads us to ask why. Why does there seem to be less selection pressure on sites of resource level 4? So that's what we're going to investigate with a mean field model to try to gain some insight into what's going on in these complex dynamics. And so the mean field model that we're going to write down is a simplification of the actual full dynamics, where instead of dealing with this full random 2D environment, we're going to make use of the fact that the resource levels are spatially uncorrelated to say, let's not worry about the actual spatial locations of the organisms. Let's just focus on how many organisms there are at sites of each resource level. So we had 5 resource levels. So we're going to define 5 states, and we're going to write down a system of differential equations for the expected number of agents at sites of each resource level. So we have a set of differential equations that looks like this. So the change in the expected number of agents at resource level K with a particular phenotype gamma, and that expected number of agents is going to change due to births into resource level K from the other resource levels. It's going to change due to hopping into resource level K by organisms coming from other resource levels. And it's also going to change due to hopping out of resource level K. And then finally, we have some weight factors W here that just represent the frequency of the different resource levels. So far, we've just been focusing on environments in which each resource level is uniformly distributed. So these WKs are just a fifth, but momentarily we'll look at some environments where the resource levels are not uniformly distributed, and some sites will be rarer than others. And we can incorporate that with the mean field model. Now, there are a few important points about this model. One is that this is explicitly a low density approximation. So we're assuming that the environment is large enough that different organisms collide with one another pretty rarely. So that essentially meaning that organisms are not blocking each other from moving around throughout the environment. Because again, there's in the full 2D environment, you can have one organism per site. And so if you have a very dense environment, some organisms might not be able to move because locations around them are all occupied already. So we're explicitly in a low density regime. And what that means is that the phenotypes are dynamically decoupled. So you'll notice, so meaning that we have a differential equation here for every possible phenotype we could have in this environment. And for the sake of handling this numerically, we're going to discretize that phenotype space. So for each hopping rate, we'll just consider 6 possible hopping rate values instead of the continuum that we can deal with in the agent-based simulations. And so what that amounts to is that we have about 7,000 possible phenotypes and then times 5 equations for each of these sites. So while the dynamics of the different phenotypes are decoupled here, they are still all coupled through this catastrophe that's going to uniformly reduce the population back down to its original size. And so we can handle this within this mean field model. And some of you may have noticed that these dynamics that I've written down here are just linear. So we could write this system of differential equations for each phenotype as a linear matrix system. So the time derivative of the vector of organisms in each state is just a matrix acting on that vector. And so this has, and again, there's one of these differential equations for every possible phenotype here. So this is just a linear system. We can solve it using a matrix exponential. Was there a question?
Oh, yeah. I think maybe you're getting to it though. I was wondering where how the catastrophes are getting modeled in the mean field equation. I thought they were averaged out or something, but something is going to happen, I guess, right now.
Right. So
Yeah,
we'll see that in just a second. So so we can formally solve these differential equations in terms of the matrix of a matrix exponential, so that the configuration of the system at time t, just before the catastrophe occurs, is just related to a matrix acting on the initial configuration of the system at time t equals 0. And so where the catastrophes come in is that every time there's a catastrophe, we're basically going to scale down the left-hand side of this equation here by this factor in the middle. So this factor that I'm drawing around excuse me, is going to be the initial condition for the L+1 epoch. And so what this equation that I've wrote down here in the middle, this is an iterative map between the configuration of the system at catastrophe L+1, dependent on what the configuration of the system was at catastrophe L. So if you look at this equation, you might notice that this looks a lot like an eigenvalue equation. And indeed, if we assume that there is a fixed point of this iterative map after a large number of catastrophes, we can in fact show that the fixed point configuration of this model is going to be an eigenvector of this matrix M, which involves the birth rates and the hopping rates. And it's the eigenvector corresponding to the largest eigenvalue of the matrix M. The matrix M appears to have only one positive eigenvalue, and so the configuration is going to be the one that corresponds to that positive eigenvalue. And again, because we have a matrix M for every one of the possible phenotypes, we have this eigenvalue for every one of the phenotypes. And so I've indicated that this eigenvalue is a function of the 5 phenotype parameters, as well as the birth rates and the resource frequencies. Excuse me. And what this phenotype represents is essentially the growth rate of organisms with that phenotype between the catastrophes. And we can use this to show that in this model, there is actually only generally a single dominant phenotype. So after a large number of catastrophes, only one of these phenotypes survives, and it's going to be the phenotype that has the largest value of this function lambda max. There is a way you can have coexistence of different phenotypes, but the only way these different phenotypes can coexist is if you have two choices of the hopping rate parameters such that they have the same value of lambda max here. And so what this means is that we've derived a kind of effective fitness function in this model. So we have a function that depends on just an arbitrary instantiation of this phenotype parameter. And if we maximize it, it predicts what the dominant phenotype is going to be in the long time limit. And it's also a function of parameters of the environment. So we can use it to investigate how changing the environment might change these optimal phenotypes. But first, we're going to compare the predictions from this mean field model to our stochastic simulations. So just to remind you, this is what the stochastic simulations gave for the most competitive phenotypes as a function of resource level in this model. And we can now check what the mean fields model gives, and it looks like this. So you can see that the stochastic model, the agent-based model, and this mean field model give different predictions for what the optimal value of gamma 4 is, the hopping rate of sites of resource level 4. And so Tong and I puzzled about this for a little bit, and what Tong did was he went and he used that effective fitness function that we derived and investigated how it changed with the birth rate in sites of resource level 5. So we can vary that birth rate from a value of 3, which is what we set the birth rate at resource level 4 to be, to higher values. And it turns out that the optimal phenotype is very sensitive to this birth rate. So the mean field theory predicts a transition in the optimal value of gamma 4 as a function of the birth rate B5. And then we predicted this on the basis of using the mean field theory, and then Tong went back and ran the agent-based simulations varying the value of this birth rate and found that that transition does indeed also occur in the full agent-based model. And this sort of result isn't unexpected. Typically, when a mean field type model predicts a transition or a bifurcation, the transition value, so the value of B at which that bifurcation occurs, is normally slightly different in the full stochastic model. But qualitatively, the mean field theory does predict correctly this sort of transition as a function of the birth rate of these highest resource sites. And that makes some intuitive sense. If the value of the birth rate at the highest resource level sites is not much higher than the birth rate in the next highest resource level sites, then there's not that much evolutionary advantage for leaving a site of the second highest resource level to try to find a site of the highest resource level. So if B5 is too small, you may as well settle for lower resource sites. Excuse me. So, this makes sense, and it demonstrates that the optimal phenotype can depend on the statistics of the environment and how birth rates vary throughout the environment. And so you might get to thinking, well, what if there may be other ways that the phenotype could change? Like, for example, what if these highest resource sites are also very rare? So that's the next thing that we investigated, was that instead of assuming an equal distribution of these different resource levels throughout the environment, what if they're distributed exponentially, so that the highest resource level sites are exponentially rarer than the lowest resource level sites? How might that impact the most competitive phenotypes? So we did that with both a modified mean field model and the original agent-based simulations, and we find that there is still a transition in the value of this most competitive hopping rate gamma 4, but that transition occurs at a much larger value of the birth rate B5. So again, B4 is still fixed at 3, but now you need a birth rate of 10 or more in order for it to be worth it to leave a site of resource level 4 in hopes of finding a site of resource level 5. And here specifically, the weight of the different resources goes as 1/2 times the resource level. So we have a parameter C that's dictating how rare the high resource level sites are. And again, this is a pretty intuitive result. If high resource sites are very rare, they need to have a much larger birth rate to compensate for their rarity. And then we can go and sweep over the rarity of sites and over the birth rate of resource level 5 sites to see or to find that there's essentially a phase diagram that shows where we expect the optimal phenotype to have a gamma 4 of 10. So it will leave sites of resource level 4, which is shown in yellow here. And then there's this region where gamma 4 is close to 0. So basically, if an organism finds a resource level 4 site, it should just stay there. And then this red dash curve is the phase boundary, which we predicted using this effective fitness function that we derived for the model. And then these white dashed lines here just correspond to the parameter ranges where we have the agent-based simulation. So this line at 0 here corresponds to the original uniform simulations, and then this line at a C equals 0.5 corresponds to the exponential example that I showed on the previous slide. And we mapped out this phase diagram using the mean field model because these agent-based simulations are actually very expensive. It can take days to do some of these simulations. So it's very computationally expensive to try to map out this whole phase plane with the simulations. But we expect the mean field theory to at least get things qualitatively correct. There may be some quantitative differences, such as a lower transition here at 0, but we expect the picture to be similar in the full agent-based model. Question in chat, mean field doesn't have anything to do with the environment being 2D. Correct. So the spatial structure of the environment is not being captured by the mean field model. And so if there were spatial correlations, for example, in the resource levels, we might need a more sophisticated mean field type model to capture the effects of those sorts of correlations. All right. So, so this is all interesting and somewhat intuitive. And so the next, but keep in mind that what's going on here is all in what I mentioned was a low density approximation. So there was a large number of available sites in the environment for organisms to find. And given the results about scarcity here, that if the high resource level sites are very scarce, we expect the optimal hopping rate, gamma 4, to go to 0. We might wonder, well, if the population were a lot more dense, such that a lot of those high resource sites are already filled up, might that create a kind of dynamic scarcity that could also change which phenotypes are most competitive in these sorts of random environments? And so that's the very last thing that I wanted to show you today is what happens if we take our environments and we modify the density, and we're going to do so by changing the size of the spatial environment that we're working with. So it's too computationally expensive to increase the number of agents. That's why we're going to decrease the size of the environment that they can navigate in. And that's effectively going to increase the density of the population. And so for the next simulation results I'm going to show you, we're going back to a uniform environment where B5 is equal to 4 in the agent-based model. And just to remind you, this is what the dynamics of the mean hopping rates for each of the different resource levels looked like in the original set of simulations. And now we're going to start decreasing the size of the environment, so increasing the density. So if we increase the density from 3%, that's 500 agents on a 128 squared grid, we increase that density to 12%, we start seeing a change in the dynamics of the average hopping rate. So, 1 to 3 still saturate at a large value. Hopping rate 5 still saturates at a kind of low value, but now gamma 4 doesn't seem to be saturating at all, or if it is saturating, it's on a time scale much longer than the 20,000 catastrophes that we've been investigating here. And there's a lot of variability in that value, suggesting potentially a coexistence of phenotypes of different gamma 4s. If we increase the density further, up to nearly 50%, so half of the sites are occupied at the beginning, then we see that now gamma 4 does saturate at a value close to 0. So now the environment is so dense that it becomes more favorable for the organisms to stay at a site of resource level 4 than try to go find a site of resource level 5, which may not be available. And then, we can push this to even extreme densities where we see similar effects, except now the time scales are getting much longer just because there's not enough room for the organisms to really move around or give birth in such high density environments, like 95% of the sites being occupied at the beginning of the simulation. We have some selection, but it's very weak because the organisms can't do much between catastrophes. So this is what those hopping rates look like as a function of the densities that we tested with the agent-based simulations. So at very low density, hopping rates tend to be high, except in the highest resource level sites. But as the density increases, we see that gamma 4 starts decreasing, is maybe kind of in the middle with some variability, possibly suggesting some coexistence before it sort of saturates close to 0 in these extreme density limits. And the last thing that we just want to take a quick look at is whether we can replicate these observations by modifying our mean field model to include some kind of effect of this density. And so we can do that by essentially adding a carrying capacity to these 5 different states. So basically, there's just going to be a factor that modulates the fraction of available space per state. And this comes about by modifying the frequency coefficient W. And so if we write down the mean field dynamics for the expected number of agents at the sites of these different resource levels, we now have these 2 new factors, which are modulating the flow of organisms into resource level K and the flow of organisms out of resource level K into the other resource levels. So a couple things to notice. This model is now both nonlinear, but it also explicitly couples the dynamics of all of the phenotypes. So the dynamics of phenotype gamma depend on all the other possible phenotypes we have in the model. So if we were to look at the full, even discretized 7,000-ish phenotypes that we looked at in the low density limit, this is a very large system of equations that we would have to try to solve. Given that in the simulations, we see that there's seems to be just 2 prominent phenotypes that survive in the long time limit, one with gamma 4 equals 10, one with gamma 4 equals 0. We're just going to investigate the mean field model using just those 2 phenotypes to see if we can replicate the simulations. And so since the only difference between these 2 phenotypes is gamma 4, let's just focus on that curve for the agent-based simulations. And so if we go and run our mean field model, what we find is qualitatively similar effects, where at low densities in this mean field model, the average hopping rate out of sites of resource level 4 is reasonably high, close to 10. And then there seems to be this transition region where the average value is somewhere in the middle, perhaps suggesting coexistence or perhaps just a very long time scale of convergence to one of these 2 limits. And then at slightly higher densities, we see that gamma 4, the mean gamma 4 essentially drops to 0, indicating that only one of these 2 phenotypes survives at long times. So the 5-state model is able to qualitatively match the agent-based simulations. And we can probe it to try to understand the dynamics. I don't really have time to dissect the dynamics of the mean field model today. I just want to make a last couple of comments about the mean field model in this high density limit. And one of the main salient features here is that in this nonlinear model, we're not able to derive some kind of effective fitness function like the lambda max that we derived in the low density limit. So there's no function that just depends on the values of gamma 1, 2, 3, 4, and 5, as opposed to the phenotypes of all of the organisms in the population. You might wonder, well, what if we tried simulating this mean field model where we just have one phenotype, and so we simulate it independently for all our different phenotypes, and then just see, did one of those phenotypes give a larger number of agents in the long time limit, and then assume that the optimal phenotype would be the one that produces the largest number of organisms. And we've tried that, and it's consistent in the regimes where it seems like we already have just one phenotype dominating the population, but it's necessarily going to miss the coexistence that seems to be happening, in this range of, in this range of densities here. So we don't have a fitness function that would predict what's going on sort of in this density regime in the model. And that's something that we want to think about going forward, and whether there's some extension of this concept that will work with possible phenotype coexistence. So I'm going to give you some of the takeaway messages of the work I just talked about, and then I want to finish the time by just talking about some possible future directions for this project. So some of the things that we saw was that the most competitive phenotypes, in some sense, learn to balance birth rates and scarcity in these random environments. So we can perhaps interpret this kind of like a learning process where the population here is learning the statistical structure of the environment. And I should note that the environment here, the statistics were fixed. They weren't changing over time, so there was no consumption, and there were no long-term changes in the statistics of the environment. And so at least under those conditions, the population in some sense learns which hopping rates are suited to the environment that these organisms exist in. And in the case of a low density environment where direct interactions between different individuals were rare, we were able to derive something like an effective fitness function for this population, which was a function that if we maximize it, would predict the dominant phenotype in the, at least asymptotically, in the limit of a large number of catastrophes. And then the very last thing we showed was that when the density of the environment is high, we can no longer necessarily ignore interactions between organisms with different phenotypes. Those interactions can lead to coexistence, or perhaps one phenotype dominating over the other, which you wouldn't necessarily expect otherwise. Excuse me. All right. So that's what we saw today. And again, I just wanted to sort of end by thinking about some future directions that we want to take this work, and challenges for investigating things like more complicated organisms. So the organisms that we investigated in the model that I talked about today were relatively simple. They had their phenotype just consisted of 5 parameters, and the sensory information that they were able to work with was just whatever the resource level was at their current location. And this is sort of a challenging problem from the perspective of modeling. In trying to understand the structure of biological organisms, we sort of have this trade-off where we can either look at very complex agents with perhaps a full neural network, or a large number of agents, but it's kind of hard to look at a large number of very complex agents. And so there's a kind of, in some sense, a complexity frontier where we've developed a lot of tools, and by we, I mean the sort of scientific community as a whole. For example, investigating things like deep networks or agents with recurrent neural networks performing some task where you can have a network with perhaps hundreds of neurons, but it's kind of difficult to investigate many agents with complete recurrent neural networks interacting and trying to understand how selective pressures shape their their neural circuitry. There is some recent work sort of straddling the complexity and large number of agents. In particular, reinforcement learning has been looking at models with many agents here, and training them with this sort of more abstract cost function approach that I mentioned at the beginning. But then if we want to go to models like I just talked about today, we can simulate a modestly large number of agents, but like the ones we saw today were relatively simple. And what we'd really like to do, going forward or long term is try to push on the complexity of these agents and perhaps simulate the competitive dynamics of agents with, perhaps a modestly sized neural network that's going to dictate how they take in information from the environment around them and use that to determine how these organisms move through their environments, and then investigate selective pressures on those organisms and whether those might align with some of the these more abstract ideas that we tend to work with in terms of cost functions. And so just to give perhaps one example of what might be a tractable problem in this, one of the things we might want, that I think would be interesting to think about is agents with multiple different kinds of sensory modalities. We're all familiar with several different kinds of senses. We have vision. We can smell. So there's all these sorts of different kinds of sensory signals in our environments, and out in the wild, organisms can use all of this different kinds of sensory information to locate regions in the environment where they will, be protected, safe, and be able to give birth to offspring. And so one question that is perhaps tractable with this sort of more population dynamics approach is, what if we have a population of organisms that have different kinds of sensory strategies, or different ways in which they try to assimilate sensory information in their environment. And those different kinds of sensory signals may have different kinds of correlations, or maybe more or less informative about the locations of high birth rate sites in the environment. And if we were to take this sort of more abstract cost function approach to trying to understand this, we might assume, as we often do in computational neuroscience, that perhaps the best thing for these organisms to do is focus on the sensory signals that maximize the information that the organisms have about where high birth rate sites in the environment are. But given the diversity of organisms that we have, we might wonder whether these sorts of evolutionary dynamics might just give rise to coexistence of many different kinds of agents, perhaps favoring different sensory modalities or combining these different sensory signals in different ways. And so this is a direction that I think or hope we could sort of extend this kind of work to try and investigate some of these problems. And this is where anybody in the IACS community that is interested in any of these sorts of problems, I would love to talk to you, because I imagine this will require some high performance computing to be able to run these simulations in a time span that is less than a graduate student PhD. So with that, let me just end by putting back up the takeaway of the work I talked about, and end by again thanking Tong Leong, my graduate student who did most of the work that I showed you today, and of course funding and support from Stony Brook University. So with that, I'm happy to take any questions in the last couple of minutes here, and of course if anyone needs to run, please feel free to go.
Yeah, thanks very much.
And thank you all for your attention.
So Braden, actually, I'm going to jump in quickly with the question, following up on your last comment, is that, could you comment on how you implemented some of these simulations and where you've run them? Because precisely trying to get at is, what's the gap between what you're doing and the possible speed that you might be able to achieve?
Yeah, so, and Tong can feel free to jump in if I misspeak or anything, but so these are, as I said, this is a set of agent-based simulations. So basically the way it works is we start with 500 agents. Each of those agents has a defined location in the environment and their own individual set of these hopping rate parameters. And then basically this is a big Gillespie type algorithm simulation. So whether an individual organism hops at any particular time or gives birth is drawn according to a continuous time Gillespie algorithm. At each.
So let me zoom in a bit more. What language is it implemented in? And
so this has been done in Python.
Oh, okay, good. Okay, good.
Yeah, and Tong has been using some of the computing clusters that we have in the Center for Neural Circuit Dynamics, and he may have made use of some of IACS's computing resources as well. And I know he's talked to a few of the students at IACS to get some tips on how to speed up some of these simulations.
So we've got a new computer and we're looking for projects to optimize on that. And this might actually be a fun thing to work on. And a lot of the optimizations that relate to this new computer would also be very, very relevant to working on GPUs. And I know that many others in the group know all about GPUs and can appreciate their speed. So let's follow up. Thanks, Braden.
Yeah, so definitely we would love to make use of more of these computing resources available to us to speed some of these things up, especially if we want more complicated agents, since I think that will dramatically increase the memory and simulation time needed.
Yeah, great.
Braden, quick question. What did you mean by Cholesky, continuous time Cholesky?
Gillespie.
Oh, Gillespie, sorry.
Yeah, sorry. Well, was that all?
Yeah.
Okay, then I think I see Margaret has their hand up.
Yeah, I put this in the chat too.
Okay.
Could some of these simulations and the sort of inner complexity explain the idea of punctuated equilibrium, which is sort of that the evolution doesn't progress along a simple line, but there are these moments where something changes dramatically, and it doesn't have to be that the environment changed dramatically in order to have something change dramatically. Just spitballing.
Yeah, I haven't thought about it in terms like that, but it's certainly an
one of the
interesting
graphs
question.
that you had with like the really stuff was really bifurcating pretty quickly. I was like, ooh, that looks like some of the graphs of punctuated equilibrium that I've seen. So it just
I see.
reminded me of
Yeah,
it.
I'd have to definitely take another look at that literature. It's been a while since I've thought about it, but I think we're talking about one of like these slides. Is this what you're referring
these like sort of massive changes that you're just sort of like, whoa, and then a steady state and then a massive change again. It just really
I'd
struck my
have
eye.
to think about it. Would be interesting to investigate whether this can happen during different time periods within a single simulation, or perhaps averaged over different trials, because this is sort of varying the initial density as a parameter that we're seeing this bifurcation. But it would certainly be interesting to see if, because the number of individuals is growing within a given time period between these catastrophes, if they can reach a certain critical density within one simulation, at which time something suddenly changes. Certainly something that bears further investigation and thinking about. Thank you for the question. Any other questions? If not, you can always shoot me an email. Braden.brinkman@stonybrook.edu. Happy to tell you more.
Okay, Braden, let's thank you one more time. And I guess